Ladies and gentlemen, please welcome Dr. Heather Evans.
This morning, it is my pleasure to introduce Dr. Ronald Maier, this year's Max Harry Weil Memorial Lecturer. Dr. Maier is the Jane and Donald D. Trunke Professor, Vice Chair of the Department of Surgery at the University of Washington, and Surgeon-in-Chief at Harborview Medical Center, the Level 1 Trauma Center in Seattle, Washington. Dr. Maier is past president of the Surgical Infection Society, past president of the American Association for the Surgery of Trauma, past president of the American Surgical Association, and is the current president of the American College of Surgeons. His interest in trauma has involved studies of acute management of the severely injured and impact of trauma system development on outcomes. Please join me in welcoming Dr. Maier as he presents Response to Injury and Stress, a Genomic Storm.
Good morning. Welcome. It is indeed a pleasure and my honor to be here to present this memorial lecture. As you know, Max Harry Weil was a driven and visionary individual. I had the honor of meeting Dr. Weil among his numerous colleagues, friends, and co-supporters. He was an amazing man. He had great insight. He had the drive to change the paradigms of how we look at cardiophysiology, how we deal with resuscitation, how we use lactate to monitor perfusion, how we use vasopressors in the shock state. He was a truly enlightened leader and paradigm changer, and it was totally appropriate when he was chosen as the first president of the Society of Critical Care Medicine. In addition, he was an outstanding mentor, friend, and colleague, and trained hundreds of subsequent critical care physicians. And for this, he should be equally remembered and recognized for his outstanding contributions to the field. He was a paradigm changer. And what I'd like to cover today is a change in the paradigm of how we who deal with the severely injured patient approach the patient and our understanding of the basic biology beneath the changes we deal with clinically. Now, this theory, which actually has become a dogma over the last 35 years, which has been through my entire career, explains what we were facing clinically and helped explain what our clinical scenarios were telling us, we thought. And that is, it was the first. It was the stimulus, the major trauma, the shock episode, the ischemia reperfusion, the burn, the multiple soft tissue injuries, which we now release damps and lead to the aggressive inflammatory response, produces a massive inflammatory immunomodulatory response, bystander injury, multiple organ failure, which then either went on to resolution or, in many cases, we thought there was a counter-regulatory response to try and down-regulate this excessive inflammatory response, leading to immune paralysis and dysfunction, which then produced a condition in these critically ill patients for susceptibility to nosocomial infection, secondary HIT phenomena, and then a heightened response of the immunoinflammatory response, worsening mods, this usually on days 5 to 7, which fit the clinical scenario, and then an increased mortality late from multiple organ failure and in some patients resolution. And as I say, this is sort of the theory that led almost to a dogma that we practiced for the last 35 years. Until several things changed that allowed us to look at this somewhat differently. We were able to map the entire human genome for the first time, so we had our roadmap to look at how genes were being processed in the genomic production of protein. Secondly, we learned how to link alterations in the genetic pattern and single nucleotide polymorphisms that we found were clinically critical in the host response to stress, infection, cancer, and many other diseases. And thus, we also found that we could just use tags because these changes and these deficiencies in the genome tended to be linked. So rather than have to investigate all million, we could use tagged SNPs and other markers that were linked to multiple other abnormalities and thus allowed us to scan the entire genome very efficiently for the first time. And then lastly, we developed the high throughput technology, the gene chips, the proteomic chips that allowed us to scan thousands of bits of information on a single small chip and to produce enormous amounts of data about the genomic state of the patient. Using this, we developed a new approach to understanding biology. Starting with the microarrays, proteomics, and the bioinformatics, we then could understand the molecular changes that were being created through these genomic changes, which then from our previous studies, understanding the individual molecules of immunomodulation, we could link to one another and create pathways that the genomic upregulation was telling us were affected. And these pathways then just led to subcellular functions in the various cells, whether it was migration, other problems, or upregulation of pathways involved in immunology or innate inflammation. These then created the phenotype of the individual cells. These are then linked together and produce the systems approach to understanding the final host phenotype and how they've responded to severe stress. So this is a story, a lot of the GLUE grant initiative that lasted essentially 10 years, was funded by the NIH GMS, involved numerous multi-specialty organizations that usually don't speak to each other. And the reason it was called the GLUE grant was because it glued together individuals and researchers from very different backgrounds to try and create an end product that was greater than the parts. And to look at the genomic response to injury, we recognized that it was unlikely we would be able to do serial biopsies of the liver, the spleen, the lung, the heart, whatever. And so we had to have a tissue that we had access to. In this case, it was the circulating white cell population, which we had known is markedly altered during periods of severe stress, and hopefully would be a surrogate for us to study the overall human response. As can be seen here, we looked at both the buffet coat, which is what most of the information I'll show you, but also there was cell separation, and I'll show you a little bit of data on the individual cell subtypes, and very little information on burn as the injury. Predominantly what we looked at was trauma patients, blunt trauma patients. I cannot extrapolate this data to penetrating trauma, to sepsis. There may be crossover, but we do not have that confirmed or do not know that for sure. These were patients who suffered blunt trauma to the point where they were in hemorrhagic shock. They had an elevated lactate, elevated base deficit, and basically required a blood transfusion within 6 hours of presenting to the hospital. If you look down below at the ISS and other problems that these patients had, they were sick. bottom line. Their ISS was high. They 30% developed multiple organ failure. They spent 10 days on a ventilator. So the mindset is they were sick. But even at that, they still only had a 16% mortality. And by the end of our study, it was already cut in half. And so we really couldn't use mortality as a binomial to try and tease out the impact of the genomics. So instead, we developed something called time to recovery from organ dysfunction, and I'll explain that a bit later. So this is the cohort. We had controls of 35 patients. The human U133 gene chip was utilized to look at the genomic response, and the results were clustered for presentation. With a false discovery rate of less than 0.001, we found that out of the 21,000 genes analyzed on these gene chips, 17,000 had a measurable change in genomic response and expression in these patients. If we cut that down to a 1.5-fold increase, it was down to 10,000. And if we did a 2-fold change in the genomic expression, there was down to over 5,000 genes could be measured as being changed in these severely injured patients. Thus, the genomic storm. 80% of the human genome changes measurably when you are hit by a cement truck. It is amazing that these patients survive at all, but the impact on their genomic function is enormous. And this shows the 5,000 genes that had a 2-fold change. And as you can see, we thought there would be predominantly upregulation, as shown by going from basically non-functional expression to upregulation, as displayed by red in this heat map. These are control column, and then it goes out to 28 days of sampling. What actually surprised us was actually there are more genes downregulated immediately after the injury at 12 hours than there was upregulation. Most of our studies had focused on TNF, IL-1, inflammatory mediators of the innate response, whereas most of the response was actually a downregulation. And if you look at the pathways that are downregulated, they're almost all involved in adaptive immunity. So as we predicted, the upregulated pathways are the innate response, the pro-inflammatory response, the causers of the bystander injury and multiple organ failure, but the majority are actually not secondary, but simultaneously downregulation of adaptive immunity, which explains why your patients in the ICU after severe injuries are very susceptible to nosocomial infections, poor wound healing, and multiple complications. And I just have to point out, if you look at this right here are the immunoglobulin responses, which, as we know, at about 7 to 10 days, we have this large uptick in production of immunoglobulins, which we really still don't know the reason for this, but you can see this very nicely on this heat map where it occurs. Comparing this to the proteomic response, just to show that this genomic response is actually producing active protein, you can see that there's a nice correlation with, again, upregulation of the pro-inflammatory innate and downregulation of the adaptive immune proteins, matching the genomic response. And then we can look at a few of the responses. This is TLR, which we know are the receptors expressed on this cell membrane that capture and regulate response to endotoxin, to DAMPs. What's interesting is they're all upregulated in these injured patients except right here. And as we're finding and re-finding, there's a upregulation or loss of control of viral infections in these patients with increase in CMV expression and others. And this is the one toll receptor which is downregulated. It's somewhat teleologic. It's not proven, but it's a nice correlation with what we see in these patients with reactivation of chronic viral disease and loss of the major mechanism that protects against viral expression. Looking at monocytes, one thing we've looked at for a long time, and I just heard in a couple lectures yesterday, we're going back to looking at the expression of HLDR and HLD, other molecules as a marker of immune suppression and predictor of outcome in the ICU after sepsis. If you look at, in these patients with trauma, this network, which is predominantly driven by interferon gamma, is totally downregulated. Blue means these proteins are not being produced normally. Again, the markers that we're now predicting as biomarkers of predictability, of course, in the ICU, can be very nicely shown at the genomic level, the reason why this is occurring, and it provides a focus for potential future therapy. It's interesting that the IL-1 receptor actually goes up, but if you take this in isolation, it's not understandable, except that if you look at the previous proteomic data, the IL-1 receptor antagonist is even more upregulated. So even though the receptor is upregulated, the antagonist is produced at greater levels and causes this systemic shutdown of the interferon pathways in the monocyte. If you look at other things that are simple, as many of us have, all of you in the ICU have noticed, certain patients, no matter how much insulin you give them, they seem to be hyperglycemic. And this was taken from a group of patients that had transfusion after severe injury. And as you can see, the insulin receptor is totally blocked or downregulated in production. So there's no receptor for insulin. So you can give as much insulin as you want to this patient. They're not going to be able to upregulate the glucose transport pathway in these cells, and you're going to remain hyperglycemic. Again, is there a way that we can alter this with specific precision medicine-directed therapies? We don't know, but we now know that it is there, it is present in these patients, and it is a potential target. A couple other markers on the cell biologic level. On the left is the healthy control of monocytes in a phagocytosis assay. What this shows is that 45 days post-injury, there's still marked downregulation of the ability to phagocytose. So those changes I showed you in the adaptive immunity pathways, including phagocytosis, are downregulated immediately after injury, and they can be measured functionally out to 2 months after the major insult. Again, I actually wonder why every patient that has a severe injury doesn't develop a nosocomial infection as a consequence of this impact on their immune system. And in fact, I think it's a great testimony to the countermeasures we've made as far as sterility, hand washing, all the other attempts we make to try and prevent secondary infection actually are able to prevent this dysfunction from being expressed as a nosocomial infection. If you look at T cell function 28 days after a severe injury, those that were not in shock actually are essentially the same as control. Those that went through ischemia reperfusion and a severe shock episode with their injury are still markedly suppressed at 28 days. So the patients we're sending home are by no means normal, even though they meet most tests to be discharged. So as I mentioned, we really couldn't use mortality as a discriminator because there was too small a number to allow us to have adequate power to look at the differential impact. So we created this concept of time to recovery from multiple organ failure. As you can see at the bottom, there are a small number who go on to death and stay dead, which is good. But then if you look at the patients that develop multiple organ failure who don't die within 28 days, there's, as you expect and as you know from the ICU, there's a group at every time point resolve their multiple organ failure as they move towards discharge and leaving the ICU. And we discovered a convenient break point at about 5 days. Those patients who get multiple organ failure and resolve it by 5 days versus those who have prolonged stays in the ICU, usually greater than 2 weeks, who do not resolve their multiple organ failure, to see if there is a correlation with the genomic response of these different groups of patients that might be used for future planning and testing. So here's the innate immune probe sets that I mentioned. These are the ones that are basically upregulated, as you can see here. And they start out downregulated, they go up, which is red on this heat map expression. And then by 28 days, they start to get back to blue, as shown here. This is uncomplicated, and these are the patients that are complicated. Again, uncomplicated, they resolve their multiple organ failure within 5 days. Complicated, they spend on average 10 to 14 days in the ICU and they have multiple complications. And if you look at this, you can't really tell a great difference just from the visual impact of these. But if you make this into a cartoon expression of the expression dynamics of these two groups, if you look at innate immunity, mean expression, look at those with complicated trajectory, those with uncomplicated, which means fast recovery, you can see that they both sustain the major hit and the major alteration in their genomic expression. The difference in the two groups is which group is able to restore homeostasis quickly and restore their genomic expression to baseline levels more effectively. This also explains why if you pick one protein here, it's not going to tell you how the patient's going to do. The patients who do well do produce as much abnormal protein as the ones who do poorly. So at this first 6-hour time period, you can't discriminate using a biomarker. We've been chasing biomarkers, as everyone in this audience knows, for 35 years. This sort of explains why it's very difficult, because those who do well and those who do poorly really respond the same way initially. The difference is what is their subsequent time course and genomic response over time. And those who do well return towards normal quickly. Those who have complications stay abnormal. Here's the upregulation of innate immunity, and here's the downregulation of the adaptive immunity genes. Again, rapid return, no complications, no return, persistent problems and complications. So we took this down with these differences now identified, looking at complicated versus uncomplicated, and we looked to see which genes were significantly different. And we came out with this panel of 63 probe sets. And as you can see on the left, is uncomplicated. They start out either downregulated or upregulated. And by 24 days or 14 days, they've gone from upregulation back towards baseline. The ones that were upregulated were downregulated and rapidly, as you can see, returned to normal functioning levels. If you look at the patients with complications, they go from downregulated to upregulated, and then they start to turn to normal, but they're still not even back to normal at 28 days. Down here it shows very nicely, these are the adaptive genes that are usually upregulated. They became downregulated, and it took not 14 days to return to normal, but 28 days they're starting to get back to normal. And that's what's displayed on these graphs. Again, Now that we've focused on only the genes that have a twofold difference between complicated and uncomplicated patients, they start to separate a bit more. But again, it's the same pattern. You don't see one gene go up and one gene go down, and that's going to be our biomarker that tells us what category we're in. They both do the same. The separation is the trajectory of the response, not the initial response. And then if you use these differences and go back to a reference control group and mark out these differences, it allows us to even further separate these two responses. And so the upper curve here are those with complications. And you can see their distance from basically a combination of controlled genes. And it is just a measure of the difference between these abnormal responses and the normal person's responses for an entire group of genes, these 63 genes. Those that go up and stay up have the complications. Those that go up but then quickly come back towards normal do not have complications. And again, these genes are the ones that are producing the IL-6, the IL-8, the IL-10, the IL-17, whichever your favorite marker is. These are the processes controlling their production. And if they, again, come back towards normal levels, then you resolve without you persist in having complications. And if you look at these 63 genes and you use their predictability to predict another group of independent patients in their genomes, you can show that the area under the curve makes this panel of 63 genes the best predictor of the patient's response to injury. It's better than ISS, it's better than multiple organ failure scores, all other scores that we use to try and predict patient trajectory is much worse than the ability of these 63 genes to predict the trajectory of the patient. Now, this has been subsequently validated prospectively. This has just been published in Annals of Surgery. And what we looked at was, again, the ability of this gene panel using a nanostring technology, which allows you to test the patient at the bedside in the ICU and have results back in 6 hours. It's another advance in our technology. It's based on the technology that was developed to monitor gene expression in breast cancer patients. It's the same machine, a nanostring created of these 63 genes rather than breast cancer genes, with a very rapid turnaround. It allows us within 6 hours to know where our patient is and which direction they're going in the ICU from a standpoint of complications. And again, doing it prospectively in 132 patients, we again were able to show that these 63 genes statistically perform better with an AUC of 0.8 over all other markers that we commonly use. And then there was a small group of patients in another genomic study from England that we compared to that was in the public domain. And again, it showed the same predictability in those patients. So using that data, using these 63 genes as a predictor of trajectory, and using a cut point as shown here, the red dots are those who have complications, the green dots get well within 5 days and do not have complications. We can put an arbitrary point on the extent of the change in these genes so that we can eliminate 55% of the patients out of this 132 we studied, keeping 45% of the patients, but capture 95% of the patients who are going to undergo complications during their hospital course. Again, this has been the holy grail of trying to find a predictive biomarker so that we can enlist in studies with potential toxicity only the patients that are going to benefit from the intervention. This also provides a biomarker, so to speak, that you can test this serially as you provide the intervention and show that the intervention actually has an impact on the genes that seem to be linked with a worse outcome. So, for example, the downregulation of the interferon gamma pathway I showed you leads to nosocomial infections. With this, you can select only patients, just like looking potentially at HL expression on the surface, enlist patients in the study giving gamma interferon that show that they have a very high propensity to developing nosocomial infections. You can then follow the patient and show that your intervention is actually reversing the gamma interferon pathway suppression and correlate it with surgical or nosocomial infections. So it's that biomarker that we've been hoping for that we can also trend to confirm that our biologic interventions are actually having an impact on the patient and their genomic response. And then just a couple to look at the duration of this. And as I showed you earlier, some cell function is deranged for up to 2 weeks that we measured. It probably goes much more. This is looking at 28 days using a principal component analysis, which again is a statistical method for lumping patients. Over here are the same genes sort of lumped in groups that move together, so there's not so many data points. And you can see they're all over here pretty much lumped together on this three-dimensional schema. If you look at the patients that had severe injury, they're all markedly different from control. And at 28 days when they're discharged, or after they're discharged, you can see that there's a group that's starting to move back towards control, whereas there's another group that doesn't move as much towards control activity. And if you look at the outcome of these patients, these light blue patients were all discharged home. The dark red patients were all sent to nursing homes because physiologically they were still unable to be sent home. So again, from the overall systems approach of the patient's phenotype, their underlying genome is mimicking and following what you're seeing clinically when you assess a patient for discharge. And so when you see a patient, you say, this patient's not going to do well at home, you know they're over here, and their genomic response is still markedly altered. So lastly, just to show you with a little bit of burn data, which we took out to a year, again, the innate and the adaptive response with suppression at 1 year after a major burn is still markedly altered. And those of you who take care of burns, particularly burned children, a year out, their metabolism is still abnormal, they have stunted growth, they have increased difficulties with wound healing, with nutritional status, and again, the genomic response to these genes is still persistently abnormal underlying this system response. So in summary of this part of our study, 80% of the human genome changes after a major injury. As I tell my residents at my age, if my genome is up near 80% altered, I want to go back and jump in front of the next truck because I know how I'm going to do in the ICU. But 5,000 genes change greater than twofold, and I have to give a disclaimer. We don't know that fold change really tells us anymore. Is 10 genes with a twofold change better than 100 genes with a 10% change? We don't know. It's just that we evolutionarily seem to think that bigger number change in a small number means more than a log-fold more change, but a very much less severe change. But we use twofold to do these studies to try and get a better understanding of the underlying biology. And 5,000 of our genes change. The change lasts a long time. Burns, they change out to a year. They say changed at least 28 days after severe injury. 8 of the 10 families most increased are innate immunity. What I and many of my colleagues have studied for the last 30 years is the TNF, the IL-1, the IL-8, the pro-inflammatory immunomodulators and markers. But unlike the previous paradigm where there was a sequential then change in downregulation of immunity, it happened simultaneously. And 9 of the 10 families most decreased are adaptive immunity, which fits. On day 5 to 7, everybody gets their pneumonia, they get their wound infection, their wounds don't heal. You are looking at the results of these genes and the adaptive immune pathways that are controlling your patient's response. It's a biological continuum. There's no white-black changes that allow you to pick a single protein, which we've proven, I might say, numerous times. If you use this time to recovery and multiple organ failure development, you get a very similar genomic pattern. Relatively few genes at twofold change is the only difference between the complicated and uncomplicated, and therefore can be made into a nanostring and allow us to use it for potential targeting and monitoring of therapeutic intervention. Now, I just want to give you a small piece of data. One of the problems with everything I've presented to you is that all our evidence is collected after the fact and we refer back to a normal population or cohort. We don't have within patient data. We don't have enough patients in a genomic database so that enough of them are going to be injured that we can compare their result after injury. But we think it's going to be a good predictor. So if it's going to be a good predictor from everything I've told you in this severely injured patient, being a surgeon, we cause trauma every day. And so is surgical trauma different than unplanned trauma? Well, this group from Florida did a very nice little study, and they did blood samples immediately before and 24 hours after patients having a transhiatal esophagectomy or a Whipple, 2 major invasive surgical procedures. And then they measured the gene expression levels before and after and looked within patient at what happened. And amazingly enough, with this new gene chip, they actually had 48,000 genes that they could survey. These major surgical procedures, they could measure a change in the genomic expression in 80% of the genes in the patients undergoing a Whipple. So from that standpoint, having a Whipple is, as some of us know, is similar to being hit by a truck. And some of the patients respond that way, but not nearly as many. And if you look at it with a twofold change where we had 5,000 genes change after severe trauma, after a major operation, 500 genes were significantly altered. Again, it's a very nice correlation. I don't have proof of direct causality, but these patients had 1/10 the genomic change, so to speak, that our severely injured patients had. Again, both downregulated and upregulated, and then they could measure those that were greater than twofold change. And if you look at the upregulated pathways, again, pro-inflammatory innate immune system upregulation. as you would expect from the inflammatory response. If you look at the downregulated, same. 9 of the 10 main pathways were adaptive immunity response. And once you start to look at this data and the pathways, it's unique information. And the one that caught my eye here in this week of our studies of immunology, as you can see here, is downregulated is the PD-1 pathways. And so we're taking patients that have cancer with a major operation, we're suppressing the PD-1 surveillance system for cancer recurrence. These type of genomic surveys may allow us to provide the precision medicine focus showing which patients are going to have risk of early recurrence because of alteration in their adaptive immunity versus those who aren't. It also can predict those who are going to have infectious complications and other problems. And so with this easy test, which you can return in 6 hours, we may be able to start using our genomic medicine to provide precision medical interventions based on what that individual patient needs and not what our population studies are trying to tell us or necessarily tell us. So each patient's gene expression had an average of 3.1-fold change in their genomic changes. These changes were not associated with the age, length of the operation, transfusion. It was basically not controlled by these iatrogenic factors. It was controlled by the patient's individual genomic response. What they did show was, again, the impact on particularly the adaptive immune system and its suppression positively correlated with infections and, as you would expect, length of stay in the ICU. So again, hopefully we will soon have a handle on which patients we need to be most aggressive with, and we can use monitoring to measure our therapeutic impact. So this is the paradigm which we have remodeled. As you see on the left, we had the 2-hit model, we had the initial major insult, the ischemia reperfusion. Then there was supposedly a subsequent suppression of this overreaction, which caused a paralysis of the immune system, leading to secondary infections like complications such as nosocomial infections. And then the second hit with an exaggerated pro-inflammatory response, leading to multiple organ failure and increased mortality. And we spent many, many years trying to keep this from happening and minimizing it, and I think we have been very successful in large part. MOF, death from MOF, has been dramatically decreased through our efforts. But if you look at the underlying biology by looking at the genomic response, you can see that the paradigm was wrong. It is not a sequential process, it is simultaneous. There is simultaneous upregulation and downregulation. This, as I mentioned, also explains why picking elevation of a certain biomarker does not discriminate between this course and this course. What discriminates is what that gene does subsequently. If it keeps going up and staying up, there's going to be a complicated course. If it rapidly returns towards normal, there will be an uncomplicated recovery, and reverse with the adaptive immune genomic response. So this is our proposed new paradigm approach and thinking of how the underlying genomic and subsequent proteomic and cellular function response to major injury progresses, and hopefully will allow us to intervene safely and effectively in the correct patients. That we will have the new targets, as I showed you. I participated in a gamma interferon trial 20 years ago, which we did poorly, and it failed, but it did what it was supposed to do from a biologic standpoint. It decreased infections. We have the genomic data confirming that. We can monitor the genomic response to gamma interferon. We can give the gamma interferon only to patients that are going to benefit from it, and again, hopefully have a major impact on outcome and the precise medical treatment that that individual patient needs. That is our goal. And it, again, will allow us to have a marker to confirm that what we're doing is doing what we think it's doing, which will also enable us to move rapidly in the future. So, outcome modification, the patient able to achieve homeostasis wins. As I always tell the residents and students, and I see Dr. Zimmerman, one of my colleagues from Seattle, a pediatric intensivist, I always tell the residents and students, I said, you know which patients are best able to achieve homeostasis and win. And I gave you the hint. It's kids. They are phenomenally durable, and their ability to homeostase and survive major insult is amazing. I don't have selective genomic responses in kids after major injury, but I would bet that they are on the curve that rapidly returns to normal after the insult. So what we hope we can give you in the near future is being able to treat the right patient with the right treatment at the right time. It's been our goal for many, many decades. Hopefully this information and this new technology that allows it to be available and timely at the bedside in the ICU will add significantly to our opportunities to improve our patients. Again, from Seattle, I thank you for this great honor and appreciate it. Thank you.